All right. I think we can go ahead and get started. 4 o'clock, I have started recording this lecture. So just want to let you know that this morning's lecture, is this too bothersome? Is there an echo or is it okay? Okay. Only because when I turn around and I'm writing on the board, I like to have the microphone on so that everybody can hear me. Okay, so I just wanted to let you know that I have posted the lecture from this morning, if you want to re-watch that. Also, I have included a link to that Kahoot game that we played, so if you want to review that too, you can take a look at it here. I think this is the first time I've actually captured every single lecture for this section, so that's awesome. I know. So super excited about that. They are all posted. And if you have any questions or have some issues accessing it, just let me know. All right, so do we just start from the beginning like we did last time? Okay, just start from the beginning. Okay. The first thing that I wanted you to take a look at is if we go to study aids for exam 2, I want you to take a look at these definitions. So terminology, as always, is, a big part of the exam. I'll just blow this up a little bit and you can kind of take a look at it. You can see that the first part of the very first lecture within this module is really all about definitions. So I'll go through what is a muscle cell, what is a motor unit, what are myofibrils, T-tubules. Knowing the anatomy of the skeletal muscle is going to be actually important, okay? So that's where we'll start off. I highly encourage you all to take a look at these definition sheets when you get home. So we're just going to start with the first lecture here, lecture 16, Intro to Skeletal Muscle. This kind of gives you an overview of some of the anatomical features of skeletal muscle, cardiac muscle, and smooth muscle, okay? The thing to notice right away is skeletal muscle and cardiac muscle are both organized as striated muscle. They have striations because they're organized in sarcomeres. Okay, so sarcomeres are the single unit of contraction. By definition, they are defined from Z-disc to Z-disc. And I'll show you that in just a little bit. But I want you to take a look at these micrographs, these images. You can actually see the striations give it kind of a striped appearance. And both skeletal muscle and cardiac muscle are striated muscle. Smooth muscle are not. So we'll get into the details a little bit later. There's 2 main types of control, voluntary and involuntary. Voluntary, you're controlling, this is the somatic nervous system, motor neurons, and muscle. Involuntary are the autonomic nervous system, okay? So that sympathetic or parasympathetic surge that you get, that's going to have effects on heart rate, as we've already seen, heart rate and stroke volume. All right, so let's get to the nitty-gritty. Motor units, another term. That is one motor neuron and all of the muscle that it innervates, all the muscles that it actually comes into contact with and causes contraction. So a motor neuron is a motor unit is a motor neuron and all of the muscle fibers that it innervates. All right, so let's look at the anatomy of the muscle. You can see this is a nice diagram of a whole muscle. And if you start to break it down and take a look you do a cross section of that muscle, you can see that it's organized in bundle of fibers. This is one bundle of fibers. And then if you look at each of these bundles, you can see that they're made up of muscle fibers. So the muscle cell is actually a muscle fiber in skeletal muscle. Okay, so the muscle cell is actually the fiber. So the fiber is actually many cells that have merged together. They call them syncytial cells. That's why skeletal muscle is multinucleated, okay? So it's a very long fiber. That is the muscle cell. It has multinucleated. It has many nuclei. All right, so if we actually blow this up, we're going to look at this box in particular. You can actually see now we're looking inside a muscle fiber or muscle cell, inside of it. Now you can actually see, the first thing you see are these cylinders. Those are the myofibrils. A lot of students think when they're looking at an exam that if I ask about the muscle cell, they say it's the myofibrils that are the muscle cell. Remember, myofibrils, these cylinders, they are organelles within the muscle cell. And they're made up of the actin and myosin. These are the contractile units. All right, so a few other structures that I want you to know. The plasma membrane of the muscle is called the sarcolemma. That's the plasma membrane. You have the transverse tubules. These are called the T-tubules. And the sarcoplasmic reticulum is analogous to the smooth ER in epithelial cells, the smooth endoplasmic reticulum. The sarcoplasmic reticulum is where all of the calcium is sequestered. All the calcium is housed. Okay? Let's see. The cytoplasm of the muscle is called the sarcoplasm. So sarco is really referring to muscle. And let's see, we've got the sarcoplasmic reticulum. The end of the sarcoplasmic reticulum that is really closely associated with the T-tubules is also called the terminal cisternae of the sarcoplasmic reticulum. So this is where all those dihydropyridine receptors are. They interact with the ryanodine receptors. We'll get to that in just a second, but remember the dihydropyridine receptors, like the game we played today, are voltage-gated calcium channels. They sense those action potentials that are traveling down the T-tubules, and they're mechanically coupled to those ryanodine receptors. So they open up the ryanodine receptors, allowing for calcium to flood into the sarcoplasm, into the intracellular space. Okay, don't want to get too ahead of the story. But, okay, so the T-tubules are sarcolemmal invaginations. This is part of the plasma membrane, or the sarcolemma. It dives deep into the muscle to make sure that action potentials can travel into that area, into that space. So it enhances action potential penetration, and it's more developed, those T-tubules, in larger, faster twitching muscles. Okay? Okay, so it's not as developed in the slow oxidative muscles. Remember, fast twitch are the fast glycolytic muscles. Okay, so I'm going to try to put a few things together here too. Sarcoplasmic reticulum stores calcium, and the terminal cisternae increases that storage. Terminal cisternae is really important in, muscles that have to react very quickly, like hummingbird wings. They have a very well-pronounced terminal cisternae. Okay, so again, a few more things about skeletal muscle. They're multinucleated. They do contain a lot of mitochondria, mostly in the slow oxidative muscle. They have a lot of myoglobin and mitochondria, less so in fast glycolytic. All right, T-tubules, myofibrils, sarcomeres. We'll go through that in just a second when we kind of zoom in on it. Sarcolemma, plasma membrane, sarcoplasm, cytoplasm of the muscle, and then sarcoplasmic reticulum is analogous to the smooth ER. So we're just kind of plowing through this. Don't hesitate to ask me questions if there's something that you want me to clarify. I'm just kind of reviewing some of this material. Hi. Yes, thanks, ma'am. She's so awesome. She's really good at grading. Okay, all right, see here. All right, so I think here's where we actually start with the sequence of events that you guys had on your muscle worksheet. So now we're going to put everything together. It's really, when you're talking about muscle contraction, it's a sequence of basically 16 steps. So we're going to go through it, okay? But if you want to review it again when you get home, take a look at that muscle worksheet. Yes?
Would this be something like an action one and then the definition, or is this something we have to recall?
Yeah, it's all multiple choice as it was before. So it's not anything, there's no short answer. You're not going to have to memorize any definitions. You'll just have to recognize the definitions. There are some multiple choice questions that pertain to this. In this case, it's not a matching exercise. It's more multiple choice questions. Yep. All right, so we're going to start with the motor neuron and what happens with the motor neuron. I want to make sure that everybody remembers the differences between graded potentials and action potentials because it applies to these excitable cells. Muscles are excitable cells too, so they have both as well. All right, so I'm going to blow this up. See if I can here. All right. So it actually starts with the action potentials traveling down the motor neuron, and that depolarization waves reaches the axon terminal end. Depolarization wave. That's enough of a depolarization to open up these voltage-gated calcium channels. Calcium floods into the axon terminal end. It promotes vesicle fusion. Remember these vesicles, like our game today, they're housing, the vesicles have a lot of acetylcholine inside. That's the neurotransmitter that's important here. Acetylcholine, the vesicles fuse with the plasma membrane, and acetylcholine is released into the synaptic cleft. Now, the receptor, I hope you all remember this, the receptor is the nicotinic acetylcholine receptors. So this is a graded potential because remember, graded potentials are usually initiated by ligand-gated receptors. So the ligand is acetylcholine. It binds to its receptor, okay? Now, this receptor is an ion channel, so it's ionotropic. Ionotropic, that means the receptor itself can conduct ions. It's a channel. Ionotropic. And this is the nicotinic acetylcholine receptors. They're ionotropic. All right, so here's where it gets a little tricky. It's a little confusing at times, but this ion channel here is a non-selective ion channel. It can conduct both sodium and potassium equally. Okay, so if you think back to the first module, the first section, if you think about the chord conductance equation, if the relative conductances for sodium and potassium are 50-50, it's going to move the membrane potential halfway in between the two equilibrium potentials. So it does depolarize the cell. Okay, that's a little confusing because it seems like it would be electroneutral, right? You wouldn't get any change in membrane potential. But remember the chord conductance equation. So that will move the membrane potential towards zero. Okay, towards zero. So let's just draw a cell here. I think the thing to remember with the explanation I just gave you, I would say that you don't really, that's not going to be on exam 2, because that was on exam 1, chord conductance equation. I think the thing to really remember here is that you do get a depolarization close to 0, okay? So, you have a motor neuron. This is actually called the end plate. This is the muscle here, so let me just, this is the motor neuron. This is the muscle. Skeletal muscle. Okay? Acetylcholine release is actually going to trigger opening of the nicotinic acetylcholine receptors. That depolarization wave, remember it's a graded potential, is going to radiate out in all directions. Okay? So you can actually see there's lots of nicotinic acetylcholine receptors. Right? That's going to depolarize the end plate, and all you need is a slight depolarization to trigger the voltage-gated sodium channels on the sarcolemma. Right? Okay? So that just needs to radiate out, and then you're going to get an action potential along the sarcolemma. Okay? Here's where the gradient potentials are. Along the sarcolemma and T-tubules, you're going to get action potentials. Okay? Does that make sense to everyone? Yeah, question.
So just like clarifying, so like that like divot where we went like last time, that's like the graded potential.
Yep, these are the graded potentials right here.
Yeah. And then that's like the action potential.
These are the action potentials.
Oh, like
yes.
how you explained it in an example.
That's right. That's right. So remember with graded potentials, they're initiated by the opening. It's a ligand-gated receptor. The depolarization wave decays with distance, just like graded potentials. That's not the same as action potentials, right? This is a cluster of voltage-gated sodium channels. Action potentials are initiated by voltage-gated sodium channels, further depolarizes the cell, and then delayed potassium channels that repolarize the cell. It's enough to trigger the next cluster of voltage-gated sodium channels, action potential. The next cluster. See how the potentials are different? They just are organized differently. They're different receptors and channels. These are voltage-gated sodium channels that initiate the action potentials. Yes?
So if you asked where the graded potentials are versus like the action potentials, is it just like motor endpoint versus sarcolemma?
Exactly. Yeah. So the graded potentials are clustered at the end plate. If I said what are end plate potentials, you would say those are graded potentials, just like the question in the game today. Along the sarcolemma and down the T-tubules, those are the action potentials. Okay. All right, so how do muscles relax? How do muscles relax? Remember, you have acetylcholine esterase that is in the synaptic cleft. That's going to break acetylcholine down into acetic acid and choline. And then it's taken back up into the cell. So acetylcholine esterase is actually a really important part of relaxation of muscle. Right? You got to get rid of those neurotransmitters in order to relax the muscle. So, pretty good? Feel pretty good? Okay, let me just, all right. This slide actually talked about what are some of the drugs that could change the events within the neuromuscular junction. The only thing that I want you to leave with this review is what we talked about with myasthenia gravis. That was one of your problem sets. Myasthenia gravis is a disease, it's an autoimmune disease where you start to produce antibodies that attack the nicotinic acetylcholine receptors. They act as antagonists. They block the response, right? They block the action of acetylcholine, which causes muscle weakness. Okay? Yes.
Can you spell that, or how would it?
Yeah, so acetylcholine esterase.
The disease, they did.
Oh, the disease. Myasthenia gravis. Myasthenia gravis.
And it acts as an antagonist.
Yeah, they're antibodies that attack the nicotinic acetylcholine receptor. And it blocks the response. So I should make it so that with myasthenia gravis, I'll make a little arrow, right? They're antibodies. I'm going to depict it as like little upside down Y's. Antibodies that attack the nicotinic acetylcholine receptor. Yeah, of course. All right, so let's keep going. Don't worry about channelopathies, like we talked about the fainting goats. Don't worry about that. Here we're just concentrating on the sequence of events, your muscle worksheet. Okay, sequence of events, muscle worksheet. So let's keep going. Yeah, sure.
On the studies week, I see that there are the practice questions.
Yeah. I can post an answer key. Thank you. I forgot to do that. Okay. Oops. And if you have any specific questions on that answer key, let me know. Or on that practice exam. Okay. So, this is the slide that I usually like to lecture from. We've already talked about the end plate. These are graded potentials. The nicotinic acetylcholine receptor are, they're basically ligand-gated receptors that are susceptible to an autoimmune disease called myasthenia gravis. All right, so action potentials then propagate. Now we're back to action potentials. Action potentials propagate along the sarcolemma, and then they dive deep into the interior of the muscle, down the T-tubules. Okay? Now, let's blow this up a little bit. Okay, so here we are here. Take a look at this. These action potentials are depolarizing the cell. That's what these plus signs are. Depolarizing the cell. These dihydropyridine receptors are voltage-gated calcium channels. So they are going to sense the change in membrane potential. There's going to be a conformational change of these voltage-gated calcium channels. And that's going to open up, right? You can see in skeletal muscle, they are physically interacting with ryanodine receptors. They are actually physically interacting. So when there's a conformational change of the dihydropyridine receptors, it's going to cause a conformational change of the ryanodine receptors. Okay? The ryanodine receptors open, calcium floods out of the sarcoplasmic reticulum and floods the sarcoplasm. Remember, that's the intracellular fluid within the muscle cell. So I'm going to just go to the next slide. You'll see here. Right? So the ryanodine receptors open, calcium floods into the intracellular space, and then you'll remember that calcium binds to, this was one of the questions, calcium binds to troponin C. Right? Troponin C. So, if you need some text, again, associated with any of this, there's several different resources for you. You can look at this slide, you can look at your textbook, you can look at your muscle worksheet that has the answers on it. All right? Okay, so let's talk about what happens now. All right, so calcium has flooded into the intracellular space, and it's going to bind to troponin C. Before we get to the next sequence of events, I just want to zoom in on those myofibrils. so that you can see the anatomy a little bit more clearly before we jump into troponency and what happens with cross-bridge cycling. So here is a myofibril. Remember, this is not the muscle cell. This is an organelle within the muscle cell. This is the contractile units. Myofibrils are made up of actin and myosin. Okay, actin and myosin. Okay, so just to clarify, the green lines here are the thin filaments, which are actin. Actin can also be called thin filaments. The thick filaments, or myosin filaments, are the red here. Myosin molecules are also called thick filaments. From Z disc to Z disc, you can see this right here, right? Z disc to Z disc. That is the sarcomere. That's a single unit of contraction. Then you have the M line. You can see here the I band. The I band is only made up of actin. The H zone is only made up of myosin. And the A band is defined from myosin head to myosin head. So there's some overlap of myosin and actin within the A band. All right, just to be clear, when muscles contract, myosin is the molecular motor. Myosin walks along the actin, pulling the Z discs closer together. And just so you know, with contraction, the I band gets smaller and disappears. The H zone gets smaller and disappears. But the A band remains unchanged. Okay? Do you want me to say that again? Pretty good? Okay. So, with muscle contraction, myosin moves along the actin, pulling the Z discs closer together. Okay? The I band gets smaller and smaller until it disappears. The H zone gets smaller and smaller until it disappears. But the A band remains unchanged. Okay? Pulling those Z discs closer together. Now with skeletal muscle, it's an all or none event. As soon as that muscle fiber is stimulated, it shortens to its shortest possible length. All or nothing. Okay, so we also, we talked about that. I'm just kind of skipping to the most important parts. Just remember these elastic elements, titin and nebulin. I had a good question in my office today asking about, okay, why with sympathetic stimulation does it increase? We already know it increases heart rate. But why does it increase stroke volume as well? That's because with sympathetic nervous stimulation, it tightens up the titin so that when, right, the sympathetic surge occurs, you get a more forceful contraction, which increases stroke volume. So the thing to remember is with sympathetic stimulation, you get an increase in heart rate, increase in stroke volume, which translates to an increase in cardiac output and mean arterial pressure. Everything goes up. All right, so titin is an important elastic element. All right, so here is what myosin looks like. Myosin has a long chain. This is a heavy chain, long tail. Light chains are on the neck. Now, I just want to remind you that light chains are very important with smooth muscle contraction. Remember that? Myosin light chain kinase. This is where the light chains are, on the neck of myosin. And then the head region has both the actin binding site and the ATP binding site. Okay. All right, I would say don't worry about tropomodulin or CAPC. That won't be on your exam. Let's look at the actin filaments now. Okay, actin filaments. The actin filaments are globular actin that actually comes together as F-actin, which is filamentous, right? Come together. Troponin is a set of molecules that are closely associated with actin. You can see actin here are the red balls, basically, that make a filament, thin filament. The troponin is made up of troponin C, that binds the calcium, troponin T, that binds tropomyosin, and troponin I, that binds both actin and troponin C. All right, for our purposes, for the exam, I'm always trying to get you just to focus. The only thing I want you to remember, like the question today, was troponin C. C is for calcium. Calcium binds to troponin C. What it does then is it moves this tropomyosin, this brown molecule right here, it's like a long necklace, moves it out of the way so actin and myosin can interact with each other. And there's about a troponin binds to tropomyosin every 40 nanometers along the actin filament. All right, so now you can imagine what happens. I'm going to skip to this particular slide. Okay. Here's where cross-bridge cycling. All right, so let's just review for a minute. Dihydropyridine receptors, they sense the action potentials coming down the T tubules. The dihydropyridine receptors are physically interacting with the ryanodine receptors. It opens up the ryanodine receptors, calcium floods into the intracellular space, and binds to troponin C. Troponin C, there's a conformational change, moves the tropomyosin out of the way, allowing for actin and myosin to now interact with each other. Now cross-bridge cycling can occur. Okay, so the bridge is actually already energized. ATP has already been broken down, hydrolyzed. All right? So on the myosin head, at that ATP binding site, what you get is ADP and inorganic phosphate. They're still attached to the myosin head. In other words, ATP has hydrolyzed to ADP and an inorganic phosphate. This has already energized the bridge. If this is the myosin head and this is the actin, now they can interact when tropomyosin moves out of the way, okay? Now, when the ADP and the inorganic phosphate dissociate, right, that's what's responsible for the power stroke, all right? That moves those Z disks closer together. All right, when that ADP and inorganic phosphate are removed, that's what's responsible for the power stroke. Yes?
Are the ADP and the inorganic phosphate just removed from the myosin head?
Yes, yes. They just, eventually, they will dissociate from the myosin head. When actin and myosin actually interact with each other, that's going to lower the affinity for ADP and the inorganic phosphate. They actually don't leave at the exact same time, but when they both are removed, that's when you get the power stroke, okay? Then, like the game we played today, it's ATP that's responsible for dissociating the myosin head from the actin filament. So you can actually see that right here. ATP is required to dissociate actin from myosin, okay? So now ATP is bound to the ATP binding site on the myosin head, and then once it hydrolyzes, it re-energizes the bridge. So once ATP is hydrolyzed to ADP and pi, inorganic phosphate, it re-energizes the bridge. Okay, that's cross-bridge cycling. So what happens is, as long as calcium is around, calcium promotes the binding of myosin and actin, right? When ADP and the inorganic phosphate dissociate from the myosin head, that causes the power stroke. ATP is responsible for dissociating actin and myosin, and then the hydrolysis of ATP re-energizes the bridge. And it just keeps ratcheting those Z discs closer and closer together.
How fast does this actually happen?
Quick, right? Yeah, it's pretty amazing when you think about muscle contraction. And even cardiac pacing, your heartbeat, right? Yeah, it's crazy. It's milliseconds. Yeah, milliseconds. I know, it's cool though. Yes?
So a little while back you mentioned that skeletal muscle is all or nothing event.
Yep.
So how does that work for partial contractions, like only moving around the fluid? Is that
Yep.
just some of them are only going?
Yes, that's exactly right. That's exactly right. That's how you should think about it. So every whole muscle is made up of multiple muscle fibers. Some are fast twitch, some are slow oxidative, some are in between, fast oxidative glycolytic. And you recruit a few muscle fibers when you, like you said, partially are picking up where it doesn't seem like it's much effort. But then as you pick up more and more weights, you're recruiting more and more muscle fibers in that whole muscle. Yeah. Yeah, great question. But once you stimulate a muscle fiber, it goes to its shortest length. Yep. All right, now remember each of these muscle fibers are going to fatigue at different times. Slow oxidative muscles don't fatigue as readily as the fast glycolytic ones. Fast glycolytic ones are very powerful, have lots of myofibrils in parallel, right? But they don't have a lot of mitochondria or myoglobin, so they fatigue very easily. Okay, so how do you feel about cross-bridge cycling? Remember we talked about rigor mortis. If there's no ATP within the cell, like what happens after death, then there's a persistent contraction because there's no ATP to actually dissociate actin from myosin. That's called rigor mortis. All right, that's actually the very first part of the lectures of muscle. Right? Again, it can be summarized in that muscle worksheet pretty easily. Right? Any questions about the sequence of events that causes muscle contraction? Pretty good? All right, so let's keep going here. Now we actually talked about skeletal muscle dynamics. Here's where we get into the 4 different relationships. We talked about the intensity force relationship, frequency force, length tension, and load velocity. So this is, this can be very difficult. Let's go through it systematically. I want to make sure that I break it down so it's very easy to understand. So go to skeletal muscle dynamics. We are on lecture 18. All right, so what we're going to concentrate on are those 4 different relationships. All right. Before we get into the 4 different relationships, you do need to know the difference between isometric and isotonic contraction. Okay? Isometric means same distance. In this case, you're not allowing the muscle to shorten. Like I said, if you walk over to the blackboard and I'm just pressing my palm against the blackboard, I can increase the force or tension. If there was a force transducer on the other side, it would measure the increase in force as I press harder and harder on the blackboard. But I'm not allowing my muscle to shorten. I'm not allowing bones to move. That's a typical isometric contraction. Iso means same, metric means distance. I'm not allowing the muscle to shorten, and I'm not moving any bone. Isotonic is different, right? Isotonic is just like picking up a 10-pound weight. The iso is the same, but the tonic is strength. So I'm not changing the weight. The weight is going to remain 10 pounds throughout the entire exercise. Tonic is strength, iso is same. So I'm referring to the weight that I'm picking up. but I'm allowing my muscle to shorten. I'm actually moving the bone. This is typical strength training. Instead of the core exercises like isometric contractions. Okay? All right, so there are different types of, here we go. There are different types of isotonic contractions. If you're shortening the muscle, that is an isotonic concentric contraction. If you're actually lengthening the muscle, it's an eccentric contraction. All right, so the first three relationships, intensity force, frequency force, and length tension, all pertain to isometric contractions. Only load velocity is an isotonic contraction. And if you ever see a graph that has tension on the y-axis and time on the x-axis, then you know you're looking at a isometric contraction. An isotonic contraction is going to have distance shortened on the y-axis. All right, so let's go ahead and talk about intensity force. And to your question, what we're talking about here is as you increase the intensity of the shock, meaning you're actually passing more and more and more current, you're recruiting more and more muscle fibers until you give it a maximum stimulus intensity where you recruit every single fiber within that whole muscle. That's how you actually know that you're at maximum stimulus intensity, because with a single twitch, you actually have saturated out. You can increase the shock value, but you won't get any more force with a single twitch. Everybody remember a single twitch? That's when the frequency is so low that you allow for contraction to occur and complete relaxation. So the firing rate is so slow that you're getting only single twitch, okay? So the top panel is the intensity force relationship. It's just saying that as you increase the intensity of that shock, you're recruiting more and more muscle fibers until you get to the point where you have a maximum stimulus intensity and you've recruited every single muscle fiber within that whole muscle. All right, so now the next one is frequency force. We're looking at the exact same diagram. This time we're looking at the bottom panel. So in the top panel, we've already determined the maximum stimulus intensity with the intensity force relationship. Now we're going to use that maximum stimulus intensity, and we're going to increase the firing rate. Instead of just a shock that gives you a single twitch, we're going to shock, right? That's the frequency force. And we're using a maximum stimulus intensity. So what you can achieve with skeletal muscle is you can get summation, and then finally tetanus. Summation and tetanus is achievable. So the question is, here's some formal definitions. Summation is a term that refers to the additional force produced as a result of an increase in stimulation frequency, firing rate. And tetanus is a state where maximum force generation is produced using both maximum stimulus intensity and a frequency of stimulation that produces no further increase in force. So you can get unfused tetanus. We've reached tetanus here because we can't achieve any more force at this frequency. And then we can also get fused tetanus. At this frequency, you can see that there's no relaxation whatsoever. And you can generate even more force at that particular frequency. So the question is, how does that occur? Well, it's not about calcium at all. Remember, with skeletal muscle, it's an all or none event. So you're not getting any increases in calcium within the intracellular space, right? It's not additional calcium. What's happening is you are actually increasing, let's see here. I lost a slide, that's weird. Okay. What you're doing is you're actually tightening up the elastic elements. So every time you shock it and you increase the frequency, you make those elastic elements tighter and tighter and tighter until finally you get to the point when you shock that muscle, the elastic elements are so tight that you get the maximum force generated. It all translates to the ends of the muscles, and it gives you the maximum force at that particular frequency. So, you guys feel pretty good about that? Okay, it's about tightening up those elastic elements, like tightening up that tighten. That gives you a more forceful contraction. Now, let me just bring it back to cardiac, right? Cardiac, the action potential in the ventricles the duration is so long that it lasts as long as the contraction itself. And that's again because you want to keep that muscle in the refractory period so that you can't get submation and tetanus. That's the last thing you want, is your heart to cramp up, right? And not give a coordinated beat. All right, so let's keep going. Length tension. This relationship talks about how you can vary the length of the muscle prior to stimulation. This is also known as preload. Talked about that today too in that review. So, anytime that you set the length of the muscle before stimulation, that's called preload. And it has everything to do with the length-tension relationship. So, how does that work? This is probably one of the easiest diagrams to start with. You can vary the length of the muscle. It can be too short. The middle one is an optimal length, or LO. And then you can also stretch that muscle out too far. This is all prior to stimulation. And basically when you finally stimulate it, you can see when the muscle is too short. We're looking at the red line. When the muscle's too short, you can't generate a whole lot of force. You can't get a whole lot of a forceful contraction. Optimal length gives you the maximum force. And then if you stretch the muscle out too far, again, the force generated wanes. You can't get as much force if the muscle is stretched out too far. The reason why is because of this, right? When you are at optimal length, number 3 here, you are getting the maximum number of actin and myosin interactions. The maximum number of actin and myosin interactions, which you can give the most forceful contraction. It's just like a tug-of-war game. The more people you have on each end of the rope, the more forceful the more stress you're going to get in the middle of the rope, right? Same idea here. The more actin and myosin interactions, the more forceful the contraction. If it's too short, like 1 and 2, the actin filaments actually overlap, diminishing the ability for myosin to actually bind, and there's no room to slide. right? The Z discs are already so close together, there's no room to go. So it can't generate a whole lot of force with contraction. If it's stretched out too far, like number 4 here, you can see that it also diminishes those actin and myosin interactions, so you can't generate the same amount of force. All right, so let's apply it to cardiac. This is again what's called preload. When the heart is at rest, the cardiac muscle is shorter than LO. Okay, it's shorter than LO. Take a look at it. It's probably a little bit in between 2 and 3. But as you start to exercise and you force more blood into the heart, more blood is forced into the ventricle, it stretches the cardiac muscle towards optimal length, maximizing those actin and myosin interactions so that you can get a more forceful contraction to compensate for that extra amount of blood. Pretty cool. Again, that's called the Frank-Starling mechanism. Okay? And it's all about the length-tension relationship, right? Preload. Preload is the end diastolic volume. It's the amount of blood that is forced into the ventricle, is setting the length prior to stimulation. All right, any questions? Pretty good? Length tension, yes?
When you're talking about lengthening towards optimal.
Yep, in the heart.
Do you mean over time?
No.
Exercise?
Yeah, so at rest, and your heart is contracting after stimulation. At rest, it is not at optimal length. It's shorter than optimal length. But when you start exercising and more blood is returned to the heart, it increases the end diastolic volume and stretches the heart muscle to LO. So now you get a more forceful contraction, and this is with exercise. You get a more forceful contraction to increase stroke volume. So you're basically compensating for that extra amount of blood that's returned. But that doesn't usually happen. It doesn't usually stretch the heart towards LO unless you're exercising. Yeah. It's pretty cool. Just remember, that's all about the length-tension relationship and preload, the idea of preload. All right, so we're doing great. There's some passive elements that come into play here. That blue line is just giving you some idea of how the recoil effect, just like a rubber band, when you stretch the muscle, those elastic elements are going to act like a rubber band. There's going to be a recoil effect, and that's why you get this increase in tension. Those are all due to the passive elements or elastic elements. The black line is what you measure in the laboratory, all right? And that's actually the sum of the red and blue line. So this is the phenomenon that you usually see in the laboratory when you're characterizing a typical skeletal muscle, when you hang it on the apparatus and you stimulate it. All right. Finally, the last one here is the load-velocity relationship. All right, so the issue is always the most confusing part for students, and that's why I want to go through this fairly slowly, is how do you generate this graph, right? This is the load-velocity graph. What it says, what it shows you, is that as you increase, this is an isotonic contraction. So as you increase the weight at the end of that muscle, that's the weight that the muscle has to work against after it's stimulated. You stimulate it and it has to pick up a 10-pound weight or a 20-pound weight. As you increase that weight, you can see the shortening velocity doesn't stay constant, it drops as you increase the weight or resistance that that muscle has to work against. Okay, so how do you generate this graph? You can see the dots here, green, orange, purple. What's happening here is you're looking, this is an isotonic contraction with essentially a single twitch. If it has a light load associated with it, it's not picking up a very heavy weight. You can see contraction and relaxation. It puts that weight back down. Okay, so the distance shortened is pretty great. All right. The orange line is an intermediate weight. You hang it on the muscle and you stimulate it. It picks up that weight, not to the same distance, and then it puts it back down. And then if you attach a very heavy weight to that muscle and you stimulate it, it just picks it up a little bit and puts it back down. All right? Yes?
When you say, like, it has a weight.
Yeah.
And it gets stimulated, what is the stimulation? Is there a natural
Yeah.
occurrence? Or is this
Yeah.
all like you're talking in a lab to?
It's in a lab, and the stimulator is actually analogous to the motor neuron. So the motor neuron is the stimulator. In this case, in the laboratory, you actually have an electrode that passes current and gives it a shock.
That's what the motor neuron does in real life.
That's what the motor neuron does, yeah. Yep. So essentially what it is an apparatus that kind of looks like this, I think, and then it has the muscle. This is the whole muscle. And you can attach different weights at the end. It has a stimulator just like a motor neuron would. You pass a current, you stimulate the muscle, and it has to work against this afterload. If it picks it up, then it puts it back down. Does that make sense? So you can do it with a light load, an intermediate load, and a heavy load. And you can see this is the graph you generated. So how do we actually calculate velocity? Right? If you look at this graph, velocity is distance over time. Right? Distance over time. So if this is distance on the y-axis and this is time on the x-axis, what we're doing is we're looking at the slope of the graph. So the green line has a pretty high slope, okay? And we can graph that here. That's the velocity. It's very much increased compared to the others. The orange line, the slope is a little bit more shallow, and we can graph it here. And then the purple line is even more shallow, okay? And that's here. And what we're doing is we're graphing now the shortening velocity versus the afterload. As you increase the weight, the velocity plummets. Does that make sense? Okay, so here's another concept that I want you to kind of realize too. Let's take the very heavy weight here. You can see the lag time before the muscle starts to pick up that weight is much greater. So the first part of any isotonic contraction is an isometric contraction. You have to generate enough force in order to pick up that weight and put it back down. So the first part of any isotonic contraction is an isometric contraction. Force is being generated, but it's not enough to cause shortening. Now, this is very much like the heart. I'm making these connections right now. Remember with ventricular systole, the muscle, the ventricles start to contract. It's building pressure, but it's not enough until the pressure in the ventricle gets higher than the pressure in the aorta. Then shortening can occur, right? So the first part of ventricular systole is an isometric contraction. Call it isovolumetric. Okay? All right. Moving along, don't worry about this slide. This is too complicated, okay? This is giving you the relationship between length, tension, and velocity. And it's cool. If you understand it, wonderful, but it won't be on your exam, okay? All right. So let's keep moving. Let's see, where are we? 5 o'clock. I want to get to cardiac, but I think things are probably coming together now. Let's finish up with muscle. I'll do this fairly quickly. We talked first about the different pathways to generate ATP. ATP generation is very important with muscle contraction. Remember, ATP binds to the myosin head. It's important with cross-bridge cycling. So we have to know how ATP is generated. In the first few seconds of any intense exercise, creatine phosphate comes into play here. There's an enzyme that will convert creatine phosphate to creatine. It takes that inorganic phosphate and it adds it directly to ATP. So that will help buffer any kind of depletion of ATP, but only in the first 6 seconds of any intense workout. All right, so I said in class, I'm not sure creatine phosphate is worth your money. We can argue it. I think it acts more as an osmolyte. Draws water into the muscle cell, making your muscles feel bigger. All right, so the ones I really want to concentrate on are the oxidative phosphorylation pathway and the glycolytic pathway. The oxidative phosphorylation pathway is the aerobic pathway. All right, it uses oxygen to make like 32 ATP molecules, 32 to 36 ATP molecules. Now, we skipped a lot of steps. If you guys were taking general biology, cell biology, maybe even biochemistry, you would know all the different steps. But with physiology, we're going to cut that all out. We've got more important things to concentrate on. The glycolytic pathway is the anaerobic pathway. I think the things to really understand here is it only generates about 2 ATP. It's not as efficient. And lactic acid is actually a byproduct. All right, so ATP is important, the depletion of ATP with fatigue. Okay, so as you deplete ATP and those potassium gradients get disrupted, extracellular potassium goes way up, especially in the T-tubules. It actually causes fatigue. And if you don't allow enough recovery time, fatigue will actually occur sooner. All right, pretty good so far. I know we're going through this pretty fast, but here's where we talked about the differences between slow oxidative and fast glycolytic. Don't concentrate on fast oxidative glycolytic, that's the intermediate one. Just concentrate on the two extremes. The real difference between slow oxidative and fast glycolytic. And that's why I really like this figure here. It shows you really the differences between the two. The red cells right here, this is a cross section. The red muscle fibers right here are rich in myoglobin, that's why they're red. Myoglobin is a molecule that stores oxygen within the muscle cell. So, rich in oxygen. You can see these black dots right here are capillaries. So, it's a rich in blood supply as well. It's bringing in oxygen, it's bringing in nutrients. It's small in diameter because it doesn't have a lot of myofibrils. So, not real strong, but they don't fatigue easily because they stay in the oxidative phosphorylation pathway. Plenty of oxygen. The opposite extreme are these orange ones. They're large in diameter, very powerful muscles. They have lots of myofibrils in parallel with each other. Right? When they're in parallel, you can get a stronger contraction. But they don't have a lot of myoglobin. They're the white meat of any animal. No, not a lot of myoglobin, not rich in blood supply. So they actually are powerful muscles, but they fatigue very easily. Okay? All right. So if you want to take a look at the differences, just concentrate on slow oxidative and fast glycolytic. And I talked about crocodiles being mainly made up of white meat. Oh my gosh! I actually saw on Facebook a guy that was talking exactly this. He was in his pool. He said if he could just wrestle it for 5 minutes, he knew it would fatigue and just poop out. So, and it was actually, they talked a little bit about slow and fast glycolytic muscles. So, cool, just exactly what I said in class. All right, and remember, each whole muscle is made up of all 3 types. Yeah?
So the fast glycolytic or the orange cells?
Yeah. So the fast glycolytic are the very light orange cells, right? You can see that. All right, and it's just as you would expect. With the fast glycolytic, they don't have a lot of mitochondria, right? That's the ATP powerhouse. Color is white, diameter is large, surrounding capillaries few, so it's everything that we talked about. If you can imagine this figure, then you can really think about it. Oh, one more thing here. This one is showing you that the difference between myofibrils in series and myofibrils in parallel. So myofibrils in series, that's sarcomeres end to end to end to end, or myofibrils end to end. They can shorten a lot more, but they can't generate a whole lot of power. Myofibrils in parallel with each other, like fast glycolytic, they don't shorten as much, but they're very powerful. Okay? They can generate more force. Okay, I would say don't worry so much about flexors and extensors. I'm actually going to skip through that. Okay? So that's not going to be on the exam. I think that we covered this in a lot of more detail in the laboratory, flexors, extensors, when we did the EMG. So that's not going to be part of the lecture here. I do want to talk about smooth muscle cells. Smooth muscle cells. I'm trying to get you all to kind of pare down some of the material to get you to focus. With this one, I think this is the diagram to star. I told one student that came up to me after class, he couldn't make it to the review session, but he really wanted me to make sure that I talked about the diagrams that would be most pertinent for the test. So I'm trying to do that here in the recording. Yes, this is one that I would star and no. Smooth muscle is not striated. It's not organized the same way. It doesn't have troponin, doesn't have tropomyosin, doesn't have sarcomeres, right? It's not organized the same way. It does have actinomyosin, and it does require an increase in intracellular calcium. Okay, so those are the similarities. All right, so an increase in calcium within the intracellular space activates an enzyme called calmodulin. Now remember, no short answer on the exam. You don't have to remember the term. You just have to recognize it in a multiple choice question. Okay? Calmodulin is activated. It is an enzyme that activates myosin light chain kinase. So remember those light chains on the neck of myosin. What that does is an activated myosin light chain kinase, you can see once you phosphorylate that neck, that's what energizes the bridge in smooth muscle. Then you can get cross-bridge interactions, right? There's no tropomyosin. It's just energizing that myosin neck causes cross-bridge cycling to occur. Usually the smooth muscle contract around tubes, like the vasculature or the intestines, something like that, or the bladder even, right? Bladder. Okay, so as long as calcium is in the intracellular space, you're going to get cross-bridge cycling. Until calcium is removed from the intracellular space, then the light chain phosphatases take over. Phosphatases are enzymes that dephosphorylate stuff. And once the myosin neck is dephosphorylated, it lays back down and actinomyosin actually can't interact. So that's a major difference between cardiac and skeletal muscle. If you want to take a look at some of the flow charts, that's great. I do want you to know the difference between single and multi-unit smooth muscle. So single unit has gap junctions. So the calcium waves actually go from cell to cell to cell, and this tissue, this smooth muscle tissue, you have a coordinated contraction. It contracts in a coordinated way. Multi-unit don't have gap junctions. All right, so let's get into the cardiac stuff. Major differences and similarities between skeletal muscle and cardiac muscle. We've talked about these terms before. The action potentials are generated by the SA node in cardiac. It originates in the muscle itself. That's why it's called myogenic. In skeletal muscle, it's neurogenic because that stimulation comes from the motor neuron. Also, major difference between the two. The dihydropyridine receptor and the ryanodine receptor. In cardiac muscle, they are not physically interacting. This again is called calcium-induced calcium release. Calcium actually can be conducted by dihydropyridine receptors. It can move through that voltage-gated calcium channel. And it's calcium that binds to the ryanodine receptor and opens up that channel. Calcium-induced calcium release. But in skeletal muscle, remember they're physically interacting. The dihydropyridine receptor is not conducting calcium. It's actually physically interacting with ryanodine receptors. Now, with cardiac muscle, remember these intercalated discs? They actually have lots of gap junctions. The depolarization wave can move from cell to cell to cell to give you a coordinated contraction of the heart. The action potentials are very different from each other. In the green at the top here, the action potential in skeletal muscle only is about 5 milliseconds. But in the ventricular myocytes, the action potential lasts as long as the contraction, again to prevent tetanus, summation, and tetanus. All right, we're moving right along here. Let's jump right into some of the concepts with circulatory systems in general. This was the intro. It's not very long. We talked about open and closed circulatory systems. How does everybody feel about the essential cardiovascular sheet, the essential calculations? Do you feel pretty good about how to calculate mean arterial pressure? Want to go through that real quick? Or do you feel pretty good about it? What's that? Pretty good? Okay. Okay.
Can you clarify?
Yeah. So, yeah, exactly. That's right. Yeah, let's do that actually. Mean arterial pressure is on the equation sheet. Let's just open that up. All right, so here's how to calculate mean arterial pressure. No worries, you're gonna have this sheet, diastolic pressure plus 1/3 systolic minus diastolic. That's at rest. Yes, so if you're exercising, it's gonna bump up from 1/3 to 1/2, right? The reason why it's 1/3 is the heart is in diastole longer than it is in systole at rest, right? Okay, so how do you calculate cardiac output? Heart rate times stroke volume. Notice that that's not on the equation sheet though. It's part of Ohm's law. Just remember cardiac output is equal to heart rate times stroke volume. So if I give you something that looks like this, PQRS, T wave, right? PQRS, T wave. The R to R value, you left with beats per minute. Okay. Stroke volume is basically the amount of blood that's forced out of the heart during the ventricular system. All right? You can calculate that by knowing something about end diastolic volume. That's the total amount of blood that's forced into the ventricle. And ejection fraction. If ejection fraction, let's just say you force about 140 mils within the ventricle, and ejection fraction is 55%, then you would multiply 140 times 0.55, right? And that'll give you the stroke volume. So if you know stroke volume and you know heart rate, you can calculate cardiac output. If you know how to calculate mean arterial pressure using systolic and diastolic pressures, then you can ultimately calculate total peripheral resistance. Because this is not on your equation sheet, but good walking around knowledge, mean arterial pressure is equal to cardiac output times total peripheral resistance. Okay? Ohm's law. So, systolic and diastolic will give you mean arterial pressure, heart rate, and stroke volume is equal to cardiac output. If you know how to calculate these two, then you can just calculate total peripheral resistance. Okay. Pretty good. Pretty good. All right. So, going back to the circulatory system, know the difference between open and closed, right? Some of the terms with the open circulatory system, the fluid is called the hemolymph. The cavity is called the hemocoel, C-O-E-L, hemocoel. Okay? And we learned a little of the comparative physiology. Fish have 2 chambers to their heart, right? They have an atrium and 1 ventricle. Amphibians have 3 chambers, 2 atria and 1 ventricle. But this is where we really wanted to concentrate. So, mammals and crocodiles and birds, they all have 4 chambered hearts. Crocodiles are more similar to birds now, we know that, than alligators, which is interesting. Alligators still have 3 chambers. So, I need you to know, here's what I need you to know. Did everyone get that sheet that I gave you today? Okay, so know the 4 chambers, the 4 valves, and a few of the vessels. And if you can label this chart, you won't have any problems. All right, so the whole acronym, to push me around, really helps you with the blood flow. T is the tricuspid. Let's just go through this real quick. I'll go through this unlabeled one if you want to go through it with me. Blood is going to return to the heart by the superior and inferior vena cava. So the superior one is the one that's more anterior towards the head. The inferior is coming up from the feet, the lower part of your body. So deoxygenated blood is returned to the heart to the right atrium. When both atria contract, blood goes from the right atrium to the right ventricle. Then, and it goes through the tricuspid or the right AV. The tricuspid is the same thing as the right atrioventricular valve. Okay? So blood goes from the right atrium through the tricuspid valve, or the right AV, into the right ventricle. Then when both ventricles contract, blood is pushed from the right ventricle through the pulmonary semilunar valve into the pulmonary artery, in blue here. Remember, it's one of the only arteries that has deoxygenated blood. Goes to the lungs, gets oxygenated, comes back through the pulmonary veins, one of the only veins that is oxygenated, goes from the pulmonary veins into the left atrium. Then once both atria contract, blood is pushed from the left atrium into the left ventricle, through the left AV, or the mitral, or the bicuspid valve. Okay, so it's pushed from the left atrium into the left ventricle. Then when both ventricles contract, it's pushed through the aortic semilunar valve into the aorta. Okay, so know the blood flow and the main anatomical parts of the heart. Here's the wall. So realize too that the pericardial sac, it's called the pericardium, surrounds the heart and acts as a buffer, like a cushion for the heart. It's made up of an outer membrane called the parietal pericardium and an inner membrane that's continuous with the epicardium called the visceral pericardium. And then there's fluid in between. That's the pericardial sac. Myocardium is the muscle, and then the inner part is called the endothelium. It's the endocardium with epithelial cells that are attached. And if you need some text when you get home, if you didn't get all that, then here's some text associated with that explanation. All right, okay. Again, in the laboratory, we had a blood lab. In lecture, you don't need to know the different blood components, okay? All right, so I'm just going to skip through that. Don't worry about memorizing white blood cells. Okay, I just wanted to give you an idea of what white blood cells are all about, but that won't be on your exam. Okay, so here's a difficult one. How are we doing on time? Okay, electrophysiology of the heart. Yay, this is one of my favorites, actually. So, electrophysiology of the heart. All right, so we had a lot of questions today. With that, I'm going to just hit the highlights to make sure everybody understands the different components. The SA node is where the electrical signal starts. And the reason why these are the pacemaker cells is because they have funny channels. They're expressing those HCN channels, which doesn't give you a nice, steady resting membrane potential. It gives you an eroding depolarization that allows the heart to get to threshold, fire, and action potential. It's all intrinsic within that SA node. And the only reason why the SA node is the pacemaker is because it depolarizes the fastest. That's the only reason. Depolarizes the fastest. And so that depolarization wave propagates from the SA node to both atria, and then it arrives at the AV node, the atrioventricular node, where the signal slows down. Now, the AV node does express funny channels, but it doesn't depolarize as fast as the SA node. So if someone has atrial fibrillation, where their atria aren't contracting, then the AV node actually takes over as the pacemaker. So you have kind of some redundancy to help make sure that blood keeps pumping. All right, So because there's not as many gap junctions in the AV node, the signal, the electrical signal, slows down quite a bit. That allows for enough time for the atria to completely contract before the electrical signal sweeps through the ventricles. So it slows down in the SA node, it slows down in the AV node, and then to the bundle of His, the left and right bundle branches, where it arrives at the apex of the heart, then sweeps up the Purkinje fibers, and then goes through the entire ventricular myocytes. Don't forget about that. It actually sweeps through the entire ventricle system, all the myocytes in the ventricle. So this is what it looks like. Starts in the SA node. This is the yellowish. Sweeps through the atria, slows down in the AV node. Now the atria completely contract before the signal sweeps through the septum by the bundle of His, the left and right bundle branches, and then into the apex, the bottom of the heart here. Up the Purkinje fibers, and then this next panel shows you it sweeps through the ventricles. And then repolarization occurs going by the same pathway. The SA node repolarizes first, then the atria repolarize, AV node, left and right bundle branches, and then the ventricles repolarize. So the take-home message here, now things should start coming together a little bit. The take-home message is P, QRS, and T wave, okay? The P wave is atrial depolarization. That was the question today. QRS is ventricular depolarization. And the reason why this blip, this wave is so large is because the ventricles have the largest muscle mass. So QRS is very large, and then the T wave is ventricular repolarization. And the questions will be very straightforward, right? T wave is ventricular repolarization. P wave is, okay? Straightforward. So don't spend a whole lot of time on those questions. Yes?
Why is T repolarization is going the same direction?
Yeah, so great question. Why do the P wave and the T wave look exactly the same? So, if we go to the boards here and we look at a heart, right? Depolarization, the depolarization wave, as it sweeps from the SA node through the atria, has a very particular direction called a vector, right? So depolarization in one direction is actually going to look at, and it's going to look the same as ventricular repolarization in the opposite direction. So the T wave is repolarization as it sweeps down and then up. So depolarization in one direction looks exactly the same as repolarization in the opposite direction. Okay? Yeah?
If the ventricular areas are, if they're bigger because the QRS is bigger, how come the T portion of the wave is bigger? Because it's with
Yeah.
waves,
So the T wave, I've seen a lot of students do ECGs. They really vary. Sometimes the T wave is small, sometimes it's large. I've read that a large T wave is because they're dehydrated. It could also be the way the student's heart is positioned in their body. It just depends on a lot of different things. So the amplitude of that wave is really hard to say it's one way or the other. Yeah, there's a lot of factors that go into the amplitude of the T wave. However, this is a really good question. Why, when you think about the time period between the QRS and T wave, where the QRS causes a disease called long QT syndrome. Makes sense. All due to a defect in a potassium channel that helps to repolarize the cell. So, that won't be on your exam, but that was a really good question. All right. So, here's what will be on your exam. You do need to know the different phases of only the ventricular action potential, the action potentials that occur in the ventricles, and the action potentials that occur in the SA node, just those two. Now, remember, action potentials are what are happening in one cell. So if I took an electrode and I impaled one cell, and you impale it and it's right in the inner leaflet of that cell, I can measure the change in membrane potential, and that's going to be the action potential. So this is what's happening in a single cell within the ventricles. 0 is the rapid depolarization phase caused by the voltage-gated sodium channels. Let me skip to this slide because this gives you the equilibrium potentials. Anytime you pull the membrane potential and it depolarizes the cell, it has to be either sodium or calcium, right? It has to be either sodium or calcium. Once those channels open, think about the cord conductance equation, it pulls the membrane voltage closer to these equilibrium potentials. So 0 is a voltage-gated sodium channel. 1 slightly hyperpolarizes the cell. That's due to a transient potassium channel. It just opens and closes really quickly. 2 is called the plateau phase, and that's due to an opening of a voltage-gated calcium channel. And then 3 is a voltage-gated potassium channel, repolarizing the cell, pulling the membrane potential closer to the equilibrium potential for potassium. And then 4 is also potassium. Okay, it makes sense when you think about it. Now that you know something about equilibrium potentials and the cord conductance equation, this makes sense. All right, so why do you have to know this? Yeah?
What's the difference between 3 and 4?
Yep. So actually 3 is, this is due to a potassium channel called long QT, LQT. It's a KV7. It's a certain subtype of potassium channels. The 4 is due to an inwardly rectifying potassium channel. It's called KIR. So they're 2 different potassium channels, but they do 2 different things. Yeah. If you took Dr. O'Grady's cell physiology course, you would dive right into all of these potassium channels. In fact, I'm just going to say this. I know we're running out of time, but 3 is actually, there's another type of potassium channel called literally the human ether agogo. It's called ether agogo because it was characterized in the 1960s, and the mutations that Drosophila flies had, they had that particular potassium channel mutation. When you put them in ether, again, this is the 60s, they look like they were go-go dancing. Yep, so to this day, they're called ether agogo potassium channels. Ether agogo. I didn't make that up, but it's a good story. Okay, so anyway, these are the potassium channels that are responsible for 3 and 4. The reason why you need to know that these are sodium, potassium, calcium, is when you get into the clinics, whether you're going to vet school or med school or nutrition, you're getting your registered dietitian certification. What I'm trying to say is, basically these are targets of the antiarrhythmic drugs, right? Antiarrhythmic drugs. They're targeting these channels. 0, lidocaine, actually targets the voltage-gated sodium channels. Verapamil, if you're in clinics, that's targeting these calcium, voltage-gated calcium channels. Amiodarone, maybe you've heard of that. That's targeting these potassium channels, okay? So once you get into the clinics and you think about action potentials, you know these antiarrhythmic drugs are actually targeting the action potential waveform to help with arrhythmias, right? Okay, so the other one that I want you to concentrate on is the SA nodal cells. Phase 4 are those funny channels. Funny channels. These are also called HCN channels. They activate at hyperpolarized voltages, the beginning of phase 4. They open at hyperpolarized voltages, and they are regulated. They're influenced by cyclic AMP. That's why it's hyperpolarization, cyclic nucleotide gated channels. Phase 0 is not the same as phase 0 in the ventricles. Phase 0 is due to a voltage-gated calcium channel. So don't get those two mixed up. And then 3, of course, is potassium channels as well. So SA node is 4 is the HCN channels, so the funny channels. 0 is voltage-gated calcium. 3 is voltage-gated potassium. All right, so all the other stuff, these are just, same idea, right? PQRS T wave, you know that. You don't need to know any of the pathophysiology, so don't worry about the blockages. What I do want to get to is the system and the parasympathetic nervous system regulate cardiac pacing. So we're going to go to this one right here. I need to clean this up again still, I promise that, but all right, so here's the pacing with sympathetic nervous system stimulation. This is a sympathetic surge, a surge of adrenaline or norepinephrine. Everybody knows that fight or flight response. What it is the neurotransmitters, you can see here, are norepinephrine. And this is innervating the SA node directly. So the SA node of the heart does have nerves associated with them. You don't need them, but they can regulate heart rate. Here's the SA node. You have the sympathetic and parasympathetic nervous system that are actually innervating the SA node. Okay? So with the sympathetic nervous system stimulation, those neurons release norepinephrine, and it binds to the beta-1 adrenergic receptors. This is a GS-coupled receptor. It increases cyclic AMP within the cell. Cyclic AMP binds directly to those HCN channels, increasing their activity. And you can see here, it increases that slope 4. So if the blue line is a normal heart rate, the green line, you can see that slope 4 increases the slope, so it reaches threshold faster, increasing the heart rate. So a sympathetic surge is going to increase heart rate by this mechanism. All right, any questions so far? Pretty good? So a parasympathetic nervous system stimulation, it makes sense that it basically activates GI, right? That makes sense. It's very elegant. The neurotransmitter is acetylcholine. Acetylcholine binds to the muscarinic acetylcholine receptors, M2. It is GI coupled. It decreases cyclic AMP in the cell, and you can see that it makes that phase 4 of the SA nodal action potential more shallow. It decreases the slope of phase 4, so it takes longer to reach threshold. Yeah, question?
Is that something your body would do when you're sleeping?
It would be something, yeah, I would say yes, because the parasympathetic is the resting and digesting. So after you eat a meal, when you're digesting food, or when you're resting, yes, parasympathetic tone is going to be higher than when you're really active or exercising. Okay? Yeah, great question. Here's the other thing too. Go back to the sympathetic. Remember we talked about beta blockers. Beta blockers are like altenolol, our blood pressure medication to block those beta adrenergic receptors. Okay, so take-home message here. This is the slide to really focus on. This incorporates everything. With the sympathetic surge, it's GS coupled, it increases cyclic AMP, increases the activity of HCN channels, increases the phase 4 depolarization rate, and increases heart rate. Now with parasympathetic, it does all the opposite. It's GI coupled, decreases cyclic AMP, decreasing the depolarization rate and heart rate, but the M2 stimulation activation also activates beta gamma subunits of the GI, which increases a potassium channel called GERK that further hyperpolarizes the cell. In the beginning of phase 4, you can see that right here. In the beginning of phase 4, you get a further hyperpolarization that then again, it takes a lot longer to reach threshold, slowing the heart rate. All right. Okay, we're gonna fly through this. Heart is a pump. All right, heart is a pump. Let's go through, I think I can go through the Wigger diagram pretty quickly here. Wigger's diagram. Now, I know there was a question on that today on the Kahoot. Let's just blow this up a little bit. All right, so we're going to concentrate on the left side of the heart. And we're going to go through the cardiac cycle. All right, so with passive filling, basically with passive filling, you're just getting blood that's flowing from the atrium into the ventricles, right? PQRS, none of the electrical stimulation has happened yet. So at this point, you know the 2 AV valves are open, right? The tricuspid and bicuspid are open, allowing for blood to flow from the atrium into both ventricles. This is passive filling during ventricular diastole, okay? Then the P wave happens, all right? The P wave happens, and now both atria then contract, and it delivers that last bit of blood into the ventricles, right? You get a little bit of a bump in pressure in both chambers, in the atria and in the ventricle. So the atria is in purple, and the ventricle is in green. That's the end of diastole. So essentially, this is the end diastolic volume when the volume of blood in the ventricle is the highest. Then the QRS occurs, and now this green line is going to go up. The electrical current sweeps through the ventricles, and now the ventricles start to contract. This, as soon as the pressure in the ventricle in green gets higher than the pressure in the atrium, that's when the AV valves shut, and that's responsible for the lub sound, the first heart sound. So as soon as this green line goes above the purple line, that's when AV valves shut, lub. Then this is the isovolumetric contraction, which is an isometric contraction. Muscle's not allowed to shorten yet. Again, this is isovolumetric because there's no change in blood volume. And then as soon as the pressure in the ventricle in green moves higher than the pressure in the aorta, that's when the semilunar valves open and blood gushes out of the ventricles. That's the ejection phase. So right here, this is where the semilunar valves open and ejection occurs. Okay? So then the blood is ejected into the aorta. We get the T wave, and now the ventricle starts to relax. As soon as the pressure in the ventricle falls below the pressure in the aorta, that's when semilunar valves shut. That's the dub. Lub is down here, dub is right here. Lub, dub. And then this is an isovolumetric relaxation. So just to make it easy, during the isovolumetric contraction and the isovolumetric relaxation, all valves are shut. No valves are open. It's not until the pressure in the ventricle falls below the pressure in the atrium, that's when the AV valves open up again and you get passive filling. Right? Does that help? Okay, so if you can go through that on your own as well, you can tell when those valves are going to be open and shut. All right, so a few other things. I think we're doing pretty well here. Whoops, that's not the right one. Question I also got was, okay, so we already talked about preload. Okay, so with preload, remember as you increase the end diastolic volume, that's the amount of blood that's forced into the ventricle, you stretch the heart towards optimal length and you can get a greater stroke volume. Here's what I was talking about with the sympathetic surge, right? With sympathetic stimulation, there we go. With sympathetic stimulation, you get tightening of the titin, so that when you actually stimulate with a sympathetic surge, this is what you're going to get. An increase in stroke volume, right? This is an increase in force development, an increase in stroke volume, and it also has effects on the circupump, phospholamban. You get a faster relaxation. So here's normal in green. The orange is sympathetic stimulation, where you get a greater contraction and a faster relaxation. So sympathetic surge not only gives you an increase in heart rate, but stroke volume as well. Okay, so we've got a few more minutes. And I think with this one, you all feel pretty good. This is the topics that we've been just covering in the last few days. Yeah. So remember, Poisson-Iso's law is giving you some idea of, it's defining the resistance. It's defining those resistance terms. So resistance is actually equal to 8. L is the length of the tube. N is viscosity. We talked about doping in class. Remember, if you make the blood more syrupy, like thicker, then you're going to increase viscosity, and that's going to increase resistance. Over pi r to the 4th. So the take-home message here is small changes in the radius of these tubes translates to huge changes in resistance. Okay? So, yeah. No calculations. Just like Fick's law, yeah, it's just the relationship between the variables. So for instance, if I said the length of the tube was, 10 millimeters, and the radius was at its smallest, then you would be able to kind of compare that with the size of other tubes, the radius and the length and the viscosity, and you would say this one is higher than this one. Yeah. So if you know, and Poisson-Iso's law is given to you, so you don't have to memorize that. And you just have to make sure that you can deduce which one is going to have the higher resistance. Okay. We talked about this pretty extensively. I mean, we could go through this entire figure, but the take-home message is, in the capillaries, the area increases dramatically. And you have to take the surface area, the area of all of the capillaries. So as the area increases, velocity actually has an inverse relationship. The velocity of the blood decreases tremendously in the capillaries to allow enough time for oxygen and nutrients to be exchanged. In series and parallel, just remember that in series, the resistance adds up. And I put this on your campus website too. So R1 plus R2 plus R3, that's going to give you the total resistance when it's in series. You have to do one after the other. But if they're in parallel, like in the pulmonary circulation, then the total resistance is less than the lowest resistor. Okay, so take a look at that canvas site. That's why the pulmonary resistance, all of those capillaries that are in parallel with each other, that's why it's so low in the pulmonary circulation. Okay, so I think you understand that relationship. Go through arteries, arterioles, capillaries, venules, and veins. Remember, the arteries are very elastic. Okay, arteries act as dampeners. There's an elastic recoil effect to keep the blood moving even when the heart is in diastole, which is pretty cool. Arterioles, don't worry about arterial sclerosis or atherosclerosis. The pathophysiology, I'm just trying to get to the core of the foundational stuff. So don't worry about the difference between arterial, that's just hardening of the arteries, arterial sclerosis. Atherosclerosis is a type of arterial sclerosis. Atherosclerosis is when you have plaque formation and hardening of the arteries. Don't worry about sphygmomanometer. We talked about that today, but that won't be on your exam. Arterioles are a dynamic site of regulation. The take-home message is if you divert blood from one organ, you have to increase flow to a different organ to maintain your pressure reservoir, your mean arterial pressure. Okay, so when you have a sympathetic surge, fight or flight response, what it does is it constricts blood to your periphery, your hands and your feet, and it diverts blood to your heart and your brain, right? In case you have to run or fight, you're diverting blood. Now, you want to make sure that you divert blood and you keep mean arterial pressure the same, that pressure reservoir. Okay, there's extrinsic and intrinsic control. We'll talk more about this when we get to the kidney. Capillaries, let me just get to this one. Today we learned about net filtration pressure. So, the take-home message here, here's the equation, you're given the equation. When you calculate it, if it's a positive number, you're going to get filtration, pushes the fluid out. If it's a negative number, you're going to get absorption. You're going to pull fluid in. And I can guarantee this will be a test question as well. The lymphatic system, one of the main roles is to return that last bit of fluid back to the circulatory system because there's a slight mismatch. You have more fluid that's forced out of the capillaries than you have fluid that's pulled in. So about 1/10 of the fluid that's forced out gets left in the interstitial fluid, and it's the lymph system that's responsible for returning it. You know that because diseases, filarial worms, that block the lymph system actually cause profound edema. This is elephantiasis. Veins act as a reservoir, and then finally, baroreceptors. Baroreceptors are detecting either low blood pressure or high blood pressure, okay? If you have very low blood pressure, let's say you're hemorrhaging or something, you've got a wound, you're losing blood, your body's going to detect the low blood pressure. It's going to send a signal to the medulla oblongata, the cardiovascular control center, and you're going to get a sympathetic surge, okay? What does that do? Increase heart rate, increase stroke volume, increase cardiac output to try to get your mean arterial pressure back up to normal because you're losing blood. Again, it was a good observation when we talked about it in class today. The trouble with that is you can bleed out faster, right? Parasympathetic, I didn't get to talk about this in class, but here's a good example of parasympathetic. This is a procedure that actually doctors use. If you actually have a blockage of the carotid artery, that's where your baroreceptor is, they will thread a balloon to that area to open it up. And what usually happens is they blow up that balloon, they open it up, and you can imagine it triggers the baroreceptor and it simulates high blood pressure. The person's heart rate plummets in the operating room or wherever they're doing the procedure. I mean to like 30 beats per minute. And they kind of have to hold their breath until it comes back up to normal. But again, it simulates high blood pressure, right? And that causes the cardiovascular control center to have this parasympathetic surge. Decreases sympathetic and really ramps up the parasympathetic, causing a profound bradycardia. Remember, bradycardia is a slowing of the heart rate. Because there's a cholesterol, a plaque in that area, so the balloon at first is just to open up that artery.
No, they don't do it on purpose to lower that.
No, they're not trying. That's just an after effect of trying to open up that artery. Yeah, yep. Oh my gosh, another marathon session. I push it right to the limit, don't I? All right, so any other questions, last minute questions? All the videos are up. I'll put the answers to the study guide or the study questions. Good luck studying, and I will see you on Wednesday. I will be dressed up, you guys. Just so you know, I'm not. I don't have any classes on Thursday.
Okay, so this is like a real life question.
Yes.
So one of my family friends had surgery on his heart, and there was stitches, and one of them popped.
Oh.
So it like filled up with blood, but why would that cause your heart to stop beating? Is it just because it can't beat?
Right. If you don't have, you can imagine. This is probably like what we talked about with the length and tension relationship. If you don't have a whole lot of blood, because the blood is actually leaking out, it's not going to stretch that muscle so that you're not going to get a forceful
So it
contraction.
completely stops it?
Pretty much.
Okay,
Yeah.
I was just wondering because it just happened like a couple days ago. So,
Oh no.
but they found him and he's all good now.
Okay.
But he went into like full cardiac arrest because of it.
Yeah, I bet.
It's crazy.
Yeah, that is crazy. So I'm sure it didn't affect, I don't know, but it may have affected the electrical currents through the heart as well. But if you're not getting blood returned to the heart, you're not going to get a heart beat.
Interesting.
Yeah. So good question. Yeah.
I was just curious. I don't know why I didn't think about it in class before, but as far as the, you say like people take like creatine for working out and stuff like that.
Yeah. So
is it more maybe about like the regenerative process of like I don't know, how do you when you actually that using of it initially in that 6 seconds?
Yeah.
Where does it go? Or like what happens?
So it gets converted to creatine phosphate to
Yeah.
creatine. Yeah.
Okay.
Then it gets actually broken down to creatinine. And
Yeah.
we're going to talk about creatinine when we get to the kidney.
Okay.
So creatinine is basically just filtered through your kidney and you pee it out.
Yeah.
Yeah. So
And do you just collect it like to get back to creatine phosphate through
No.
just normal?
your body does make some creatine, but if you're taking supplements, you're going to have way more creatine than you normally would produce.
Okay.
But the breakdown product is creatinine, and it goes out through your kidney.
Okay.
Yeah.
Yeah.
But I can, I understand for, I think, an elite athlete where you really have like a sprint or jumping and you have a huge depletion of ATP.
Yeah.
Like in a matter of like seconds, then creatine phosphate is really going to help you out.
Even though it would still be a 6 second increment?
Yeah,
Can
because
you increase
imagine.
that increment at all?
I don't know. I have to jump into the literature to see if there is something that you can do.
Yeah.
But I know it's really important for like when you need a fast start or jumping.
Yeah.
Right? Because you're using that fast glycolytic muscle. Yes, they're powerful, but yeah, the depletion of ATP is profound.
Mm-hmm.
I think then your cell starts taking over, and as long as you're fit, you should stay in that oxidative phosphorylation pathway. And I've heard, when I think about it, it's fascinating because you always want to push yourself until you get to fatigue.
Mm-hmm.
Because then once you get to fatigue, that's when you really get a lot of those micro tears
Tears,
and
yeah. Mhm.
and you're building up those myofibrils.
Mhm.
So, and when you think about being aerobically fit, your heart is able to pump enough oxygen to the tissue that need it.
Mhm.
When you're not aerobically fit, you're not getting enough oxygen to the tissue, and it switches over to the glycolytic pathway. That's why you feel that burn, that's why you're building up lactic acid.
Lactic acid.
Yeah.
Okay, yeah.
So, if you find some articles on creatine, I will happily change my mind on creatine phosphate.
Yeah,
But
I've never been a huge, but I know buddies that are. and
some
it
people
just-
swear by
Yeah,
it.
I know. Yeah.
Yeah.
And some of the stuff that I've looked up has kind of said the same thing, where it's like it's not really like doing
Yeah.
a huge amount. And then you said that, so I was like, okay, that confirms it, but maybe I'll look into it. a little
Yeah,
more.
I do know that I think it acts as an osmolyte.
Yeah, and I totally agree with that.
It
yeah.
just
and
pulls water in.
That's what a lot of the documents say about it. Is just like you're kind of stuck on it once you're.
I know.
Once you stop.
Because once you
then
stop-
it all drains out.
Yeah,
Yeah, exactly.
exactly.
Which is another reason I'd be like, don't.
Right, don't. Yeah, it's weird. I think it's just the expense. I don't think it really will harm you.
No,
But
yeah.
I think $50 for every one of those huge powder things is
Isn't
just.
there usually a bunch of, well, I don't know, but a bunch of caffeine and other things in
That's
there,
probably
that
true
can-
too.
that can-
Yeah, now caffeine is, I swear by caffeine, and I think it enhances any kind of workout.
Are there harmful effects of too much caffeine with the heart?
Oh,
as
yeah.
far as the heart goes?
Yeah, and there's a lot of diseases, inherent diseases that could really have, like malignant hyperthermia. Caffeine also has effects on the ryanodine receptors that we've been talking about. So yeah, people that have malignant hyperthermia are very sensitive to caffeine. So yeah, you can have some underlying issues that could really put your heart at risk.
Yeah.
Hi.
So I had a few questions.
Yes.
My first question was tropomyosin on the
Yes.
smooth muscle cells. So
No tropomyosin in smooth muscle.
why does it look like there is on the diagram?
Oh.
Like that, wrap around.
In
like,
smooth
structure?
muscle?
Yeah, I was looking on the slides.
Yeah.
And then I Googled it, and some people are saying there is tropomyosin.
If there is, it's very minimal. Yeah,
Okay.
I would say there's no troponin or tropomyosin. We're talking smooth muscle, right? Yes.
Yeah. Uh-huh.
Yeah, no.
Okay, just-
Actinomycin for sure.
Uh-huh.
So actinomycin, they do interact with each other,
Yep.
and they cause the smooth muscle to kind of wrap around
Okay.
tubes, like
Okay.
the vasculature.
Mhm.
But no, I would
No?
say it's
Okay.
not organized the same way in sarcomeres or,
Okay.
tropomyosin
Okay.
or troponin.
Okay, thanks.
And there are some SR, but it's a very minimal SR as well. So most of the
Okay.
calcium is either, a lot of it is coming from the extracellular fluid.
Okay.
Yeah.
That makes sense.
Yeah.
And then for the funny channels in the heart, where is that located? Like, how does it look on the SA node? Is it on the membrane?
Yeah,
Okay.
it's actually on the membrane. It's like
Okay.
any kind of ion channel.
okay.
It's expressed on the membrane, and when it opens, it allows for both sodium and potassium to leave. So it slowly depolarizes the cell, moves it towards zero until you reach threshold,
Okay.
and you activate the voltage-gated calcium channel.
Okay.
So those HCM channels, they just slowly depolarize the cell. And basically they're the pacemaker cells only because they depolarize the cells the fastest.
Okay.
Yeah, and that's your heart rate.
Okay.
Yeah.
And then when it's depolarizing, you said that's the P wave.
Yeah, so when it is so the first part of the P wave is the SA node action potentials. And so the entire SA node, all the atria, and part of the AV node is constituted in the P wave.
Okay.
So remember the SA node, when you see an action potential, that's just what's happening in one cell
Okay.
in that patch. But the P wave, you kind of are zooming out and you're looking at the heart as a whole organ, and you're watching the depolarization wave go from cell to cell to cell to cell in a very particular direction.
Okay.
And the P wave is atrial depolarization as it sweeps through the atria in a very specific direction or vector.
Okay.
And it'll change whether you're talking about a 3 electrode leads or 12 right? If you went to the hospital, they'd put 12 electrodes on you, and they would take a picture of the heart as it rotates around the heart, so they can get a lot more information.
Okay.
But it's looking at the depolarization wave in very specific vectors,
Okay.
directions.
Okay.
Yeah.
So with those waves, I'm getting confused because
Yeah.
for like sodium and potassium channels, we've always like used those waves to depict like what the value is, like plus 70 minus.
Yeah.
So is it the same here, or is
Yeah.
it just contraction?
Oh, I see. So the electrical currents
Uh-huh.
depolarize the cell and repolarize.
Uh-huh.
And just like with skeletal muscle,
Mm-hmm.
graded potentials and action potentials happen first.
Mm-hmm.
And then calcium is released, and then contraction.
Mm-hmm.
So that's the excitation is the electrical currents, and then contraction. There's a little bit of a lag time.
Okay.
And the lag time is due to calcium being released,
Mm-hmm.
binding to troponin, moving the tropomyosin,
Mm-hmm.
right? And then
Yeah.
allowing for the cross bridges to interact with each other.
Okay.
Okay, so when we talk about the electrophysiology of the heart, we're specifically talking about the depolarization of the cell,
Mm-hmm.
and then how it's transmitted from cell to cell to cell
Right.
as it sweeps through the heart,
Mm-hmm.
and then contraction occurs.
Okay.
Right.
And
So the excitation happens first, and then contraction.
okay, so that bump for the P wave is that depolarization event.
Yes.
And then why is there still a bump for the T wave? If it's repolarization.
Yeah.
Wouldn't
So
it
that
then-
was the question that gentleman asked.
Mm-hmm.
So depolarization as it goes from cell to cell to cell in a very specific direction looks exactly like repolarization in the opposite direction.
about values? Like, if you could put a
Yeah, okay. So here's what the value would look like. I see the different places. Yeah. So the value is going to be within this cell, right? We're talking about depolarization, so it would look like this, right? And then repolarization. Okay. Then once it depolarizes, the next cell is going to depolarize and repolarize. Next cell, depolarize, repolarize. Okay. So you can see the depolarization wave is going to go from cell to cell to cell first, and then the repolarization wave is actually going to follow the same path.
Okay.
It's going to just be a little bit of a lag time.
Okay. You said the repolarization will still go up again?
So this is depolarization,
Right.
and then this is repolarization.
Right.
Yeah.
Uh-huh. So then how do you explain that T wave for the repolarization?
Yeah, so the T wave, this is already depolarized. It's gone depolarized, depolarized, repolarized, repolarized. Does that make sense? So each cell in this area is going to depolarize and then repolarize.
Uh-huh.
Depolarize, repolarize.
Okay.
And then this is going to, you're going up, depolarize, repolarize, depolarize, repolarize, depolarize, repolarize. So what you're capturing, and this is quick, is depolarization and then repolarization.
Okay.
Right.
Yeah.
So you're capturing the P wave is depolarization, and then the repolarization is masked by the QRS, the repolarization of the atria. And then you're getting depolarization and repolarization.
Oh, which is all in one curve.
Pretty much.
Oh, and that's why you're saying to look at the big picture.
Yeah.
Because it's not just, okay, one specific, like, side.
Yeah, it's not one specific side, it's measuring.
Okay.
Yeah.
Okay.
But with the three leads, you're getting very much of a two-dimensional picture of your heart. Yeah.
Okay.
So you get depolarization and repolarization.
Okay, that makes sense.
It's hard.
Yeah, that is.
Yes.
That's very hard.
And I'm glad you're really thinking about it that way too, because, yeah, we have been talking about the depolarization wave, and then you almost think that there's a couple of seconds, and then it repolarizes.
Exactly.
Yeah.
It's that slow.
Yeah, but it's depolarization, repolarization.
Okay.
Depolarization, repolarization.
Okay.
Yeah.
Makes sense.
And it's about 250 milliseconds between
Okay.
the depolarization and the
Oh,
repolarization.
wow. Okay. And
Yeah.
then my last question
Sure.
is during the refractory period,
So you get depolarization, and then it inactivates, and then that's kind of where the calcium channel takes over. So you get a slight dip with the repolarization with a potassium channel. And then the plateau phase is definitely the calcium channel. Keeps it depolarized.
Potassium?
So yeah, and then potassium starts to turn on here. So calcium shuts off and potassium turns on and repolarizes the cell.
Do they ever go at the same time?
Well, see, they don't because the delayed potassium channel actually opens with depolarization. And it's called delayed because it goes through a closed state and then it opens.
Oh, I see.
So it's, I mean, when you think about the evolution of that. Yeah, so you just have these channels that open up in sequence that causes that action potential to be formed. And that's what's responsible for the electrical current that then initiates the contraction. Yeah.
And then calcium is coming into the cell, right?
Calcium is with the cardiac muscle, a lot of calcium does come in from the extracellular side.
Wouldn't that repolarize it then? Or depolarize it, I mean?
Okay, I see where you're going. So the electrical current actually happens first. And then you're right, it's going to depolarize. It's going to increase calcium within the cell. But remember, it's the calcium movement across the membrane that's actually going to be responsible for the membrane potentials. Yeah.
So it's the calcium. So is it from inside to outside then?
It is from, there's a sodium calcium exchanger that moves calcium back out of the cell.
And that explains that, the dip.
Well, actually, this dip is turning on a very quick potassium channel that pulls it down slightly. Yeah.
Makes sense.
It's a KB 4.3 or something like that. It's a transient potassium channel. So this is all your questions are actually way beyond this class.
Oh, really?
But it's really good to kind of think about this in a more sophisticated way because then I think when you answer the questions, it'll be easy. Because the questions really are going to be things like, what's the P wave? Atrial depolarization. You know what I mean? It's going to be pretty straightforward. Nothing real tricky.
I like understanding it at this level.
Yeah, no, you're doing great. Yeah, these are